in natural populations. Now, evolution is traditionally considered to be a very slow, gradual process of small incremental changes over a very long time scale. So it's something that we can infer, but we can't necessarily observe in real time. And Darwin famously developed his theory of evolution by natural selection while a naturalist on the HMS Beagle. And when he returned to London, he gave his various bird specimens to the ornithologist John Gould, who then came back to Darwin and said, these birds that you labeled as various finches and wrens and sparrows are all actually different species of finches. And you just have them labeled as from the Galapagos, but which specific island did they come from? And Darwin had a moment that unfortunately I think we can all relate to in our research, which is, oh shoot, why didn't I take more detailed notes? But fortunately, Captain Fitzroy had very meticulous notes on his own specimens, and through that they were able to reconstruct that each of the different species originated on different islands. So I really like this example for a few reasons. First, because it shows that different selection pressures over a spatial scale can result in local adaptation. And the other thing it highlights is that there are that evolutionary processes can be occurring over a spatial scale that we assume are constant. I guess finally it also has it emphasizes the importance of keeping a really good lab notebook. Now Peter and Rosemary Grant have dedicated their career to studying these same finch population, and they've measured it over about 50 years now, which is about 50 finch generations. And they've shown that these populations are changing not only over space, but over time. And so you can see that I'm not going to try to do this point here. Okay, so you can see that there can be, the populations can shift in response to a single environmental event, like this severe drought in 1977, that can drastically, the population can drastically respond to that sort of event. So again, we're seeing that evolutionary processes are occurring on an ecological timescale. And so together, these finch populations are showing that selection occurs over space and time, and that you can't make assumptions that populations are constant. But one thing about the populations is it's looking at very discrete boundaries. So we're looking at patchiness of different islands or a very severe catastrophic weather event. And so we still don't have a very good understanding about the rate at which evolution occurs in a more continuous environment. And one of the most fundamental ways that organisms experience different environments is the changing of climate across seasonal time. Now, arthropods and other insects or other organisms that reproduce multiple times annually are experiencing different environmental selection pressures subsequently each generation. So to look at this, I'm going to use Drosophila melanogaster, which originated in sub-Saharan Africa and have subsequently spread worldwide. So they've moved from a tropical environment and have come to inhabit a variety of different climates, including temperate and very cool environments. And numerous Drosophila biologists over the past few decades have studied both genes and traits that vary along these environmental gradients to look at the association between genotype, phenotype, and the environment that are driven by natural selection and therefore reflect the adaptive process. And so they've looked at latitudinal crimes worldwide, and there's a general pattern that at low latitudes, so in tropical environments, there's an increase in reproductive output. Whereas when you increase in latitude, going north or south, depending on which hemisphere you're in, there's a general increase in stress resistance. So this really encapsulates this classic life history trade-off between reproduction and stress resistance. But it's not just the absolute temperature that is changing in these different environments, but it's the amount of change across seasonal time. And so you can see here that the flies originate in an environment that has very little change across seasonal time, and they now inhabit some locations that have strong annual fluctuations. And so I'm going to use what we know about adaptation across latitudinal climes to make predictions about what we expect to see across seasonal time. And people have frequently looked at parallels across space and found that there's similar selection pressures at high altitudes and latitudes, again, an increase in stress resistance. So we would assume that the similar environments would also select for the same sort of traits in a single location across the season. And Dobzhansky actually collected pseudo-obscura across seasonal time and altitudes, and he saw that there were parallels in frequency of chromosomal inversions. So this is showing a genetic signature of selection that something is driving this consistent fluctuation in these different environments, and therefore suggests that there's some sort of trait or phenotype that is under selection in these different environments. So again, I'm using what we know about adaptation over space to make predictions, and we would therefore predict that the cold environment of the winter would select for stress resistance. So when you collect flies in the spring, they would have elevated stress resistance. Throughout the summer, when the population is expanding and there's a release from the selection pressure, we would expect a decline in stress resistance, and therefore flies sampled in the autumn would expect it to have lower stress resistance. And because the seasons continue to cycle, we would expect an oscillation in these traits over time as well. Now my collaborators, Alan Berkland and Dmitry Petrov, have done whole genome resequencing on my samples, and we see that there are hundreds of SNPs that are annually cycling in frequency from spring to fall. So this is somewhat equivalent to the chromosomal inversions that we saw, that Dobzhansky saw, which is showing there's a genetic signature of change, and therefore it's really exciting. We can potentially look at what specific traits are being selected upon. So overall, we see that the environment is cycling across seasonal time and imposing different selection pressures, and that there's a genetic basis of change. And what I'll be talking about today is asking what phenotypes are under selection over the same time scale. And so to do this, I focused on an orchard in a temperate location in Pennsylvania, and I collected across seasonal time. I collected females that have been inseminated in the wild and established isofemale lines. So I put one female in each vial and let her lay eggs, and then maintained those lines in the laboratory for 4 generations. So by keeping them in this common laboratory environment, we remove any sort of environmental or maternal effects, and therefore when I assayed these phenotypes, it's reflecting just the genetic basis of phenotypic change. And I assayed a variety of different life history traits, and what I saw is that there's elevated stress resistance in the spring. So this is showing males and females separately, but you'll see the pattern's the same. Females are in black and males are in white. So there's an increase in, or there's higher starvation resistance. The flies are recovering from chill coma faster. They're withstanding heat stress for longer. They're also developing faster. And so this is showing across 1 year. And as I said, we would predict this pattern to cycle across many years. And I've just finished the 5th year of my PhD, and so I have 5 years of data, which shows that for the most part, these patterns are cycling in a really repeatable way. There are some annual fluctuations, and definitely the magnitude of change differs from year to year. But we also experience very different environmental selection pressures across that same time scale. So some of these winters we had 2 polar vortexes that were probably much more extreme selection pressures, and some of them we barely needed to take out our down jackets. And what I don't have time to present now is looking more at specific environmental correlations with these traits. But something that I need to note is your immediate thought is this pattern could be this rapid adaptation, but it could also be driven by migration. But when we look at the genetic level, migration is insufficient to explain the allele frequency changes we're seeing. So we can infer that that's not what's driving these phenotypic changes either. And there's also another experiment that I will get to in a little bit. Another problem inherently with measuring over temporal scales is that by standardizing the amount of time in laboratory culture before phenotyping them, I'm not able to measure these traits at the same time. So to address that, I maintained the isofemale lines in the lab, and after all the collections were complete, I reconstructed populations from each collection by allowing 40 of the different lines to to recombine in a population cage. So essentially recreating the past 4 years of change, and then I'm able to measure these phenotypes simultaneously. And so what we see is these patterns persist over time, even though they've been in the lab for a different number of generations. So overall, what I see is in this Pennsylvania orchard, there's really pronounced and rapid seasonal change in phenotypes. But the question is, how universal is this? Is this just unique to Winthilla orchards? And so to address that, in 2 years, I additionally sampled equal distance north and south. I followed the same collection regime that I described earlier. And so this is the initial figure I showed you, but I've added on the Massachusetts response in blue and the Virginia response in red. So you can see that there's parallel response across seasonal time within this geographic range, but also that the magnitude, these are all traits that are shown to be clinal on the east coast of the US. And the magnitude of change that's experienced across seasonal time is enormous compared to what is seen among the populations. So it shows how important, over the scale of 10 to 15 generations, this rapid selection response can be. So finally, to put this magnitude of change in the context of an even broader scale of a whole climb, I'm going to show you some of the same data, but just looking at the degree of difference across spring to fall. So I'm going to show you a figure that looks at the magnitude of change in each of these natural populations from spring to fall, so in a paired way. I'm going to look at the magnitude of change in those reconstructed populations that have been in the lab but were assayed at the same time. And then also in this experimental mesocosm, which is a peach tree that's encased by a 6-foot-cube cage. So it's a closed environment, and we had 6 replicate cages in Pennsylvania. At the beginning of the spring, we released 1,000 flies with a spring genotype, and then we allowed them to respond to the environment as it changed across the season in a closed system. So this is, again, addressing the issue of migration. So I'm going to compare the magnitude of change for all these seasonal measurements to what we see from Maine to Florida. And so that's what's shown here, the magnitude of change for the different traits is on the y-axis. We can see that for all of the traits in the natural populations, there's a wide variation in change, but overall it's very high. It's lower in the reconstructed populations, and that may, to some degree, affect adaptation, the release selection pressure of being in the lab. In the natural mesocosms, again, there's a lot of variation, but it's high. And all of this is showing that, all 3 of these are showing that seasonal change is equal to or greater than what's experienced over this very broad time scale, or not time scale, spatial scale. And so just in conclusion, we're seeing the populations are responding in a very rapid way to their environment. And it's really, it really can apply to all of our systems that we make assumptions about things that are being constant when this is over the scale of just several generations. And other things that I am working on are taking advantage of the system to really combine what we're seeing in terms of the genetic change and the specific environmental selection parameters. And I just have a lot of other projects related to this that I'd love to talk about during the coffee break. So briefly, I just again like to thank my collaborators. Alan and Dimitri did a lot of the sequencing and just given great advice. And my advisor, Paul, all of the members of the Schmidt Lab, the orchards that let me collect their flies, and my various funding sources. Thank you.
So, did you look, is there any degree of, so those are presumably genetic changes, is there a degree of plasticity too? Because essentially seasonal change is like what you'd expect plasticity, not evolution. It's like very predictable, right? Mm-hmm. So why do you think such strong genetic?
Well, so I haven't measured a plastic response in the field, but I actually have looked at the degree of plasticity in terms of these traits across seasonal time, and even the magnitude of change that is occurring vary when we're in different thermal environments, that changes across the time as well. So I think it's a combination. I think there definitely is probably a plastic response to different environmental conditions, and I haven't tested that. But I think there's also a very strong genetic response based on the strong selection and differential survival and reproduction.
You considered whether photoperiod is impacting you rather than temperature?
Well, so definitely photoperiod is important for some traits. I haven't tested it in response to these, but I guess I presented this in terms of temperature, but really it's in terms of whole climatic variables that are changing, and definitely shorter photoperiod and cooler temperature are tied together. We've looked at photoperiod in terms of diapause, of course, but not in terms of these traits. Well, let's get coffee.